introduction of Dr. Randall Mynatt today. He's the Douglas L. Manship Senior Endowed Professor in Diabetes and directs the Transgenic Core here at Bennington. He received his PhD at the University of Tennessee in nutrition sciences and has over 2 decades of experience in research on obesity and diabetes. He's credited with having generated over 50 mouse transgenic amniotic lines. You're going to hear about some of that work today. His research is on lipid metabolism with a focus on the regulation of fatty acid oxidation and the role of L-carnitine in moving acyl moieties across mitochondrial membranes. His lab has reported that dietary carnitine supplementation improved insulin sensitivity in 3 mouse models of impaired insulin action, including aging, genetic diabetes, and high-fat feeding. In addition to improved insulin sensitivity, they observed increases in cellular export and excretion of lipotoxic metabolites. His lab's work with carnitine acyltransferase in the mouse model has led to the finding that insulin resistance in the mouse is not secondary to obesity, but rather suggests a direct role of the carnitine acyltransferase in muscle for glucose homeostasis, therefore supporting the role of CRAT as a key enzyme in mitochondrial energy homeostasis. And I think that's what we're going to be hearing about today.
So, some of it.
Okay, so I'll turn it over to you. Thank you.
All right. Thanks for coming. So today, what I'm going to talk about is really in manipulating genes in the mouse genome. We'll talk about our different types of transgenes, how you make a transgene, the pronuclear injection process, and then I'll give you some examples of each of those mice that we've made. So before we really get started, is it okay if I stand here? Can people see? All right.
Is it not working? Oh, okay.
That's okay. I'll see if I can do this now. There it is. Okay. So anyway, these are different stages of embryogenesis. Shortly after the egg is fertilized,
The arrow keys, maybe?
I'm just going to use the, do I have a sheet of paper?
No, I need time.
That'll work. So anyway, shortly after the egg is fertilized, you have your male and female pronuclei. And then at about 1.5 days, the pronuclei fuse together to form your nucleus, and then by about day 2, you have 2 cells, and then you have 8 cells, then you have 16, and so on, until you get to your blastocyst stage on the end. And that's, so for our pronuclear injections, we do injections in the fertilized egg before it splits, okay? So you have both pronuclei, but they haven't fused together yet. And then for the ES cell injections later on, we use the blastocyst. I'll go into more detail. Okay, what is a transgene? So basically simple, you have a promoter. It can be a promoter that expresses in all tissues. It can be a tissue-specific promoter. It can be an inducible promoter. Then you have your gene of interest, right? Okay. And then you have a polyadenylation signal. And then you'll do a restriction digest and purify your promoter, your gene of interest, and the polyadenylation signal away from the plasmid backbone. and then do your pronuclear injection. So what is pronuclear injection? So we superovulate the females, and we do that with pregnant mare serum gonadotropin, and that mimics follicle stimulating hormone. And then about 46 hours later, you hit them with HCG, which mimics luteinizing hormone and triggers ovulation at a specific time. And at that point, we can collect the eggs the next morning when they're at the right stage, and they're fertilized but not two-cell. So then we remove the ovaries and collect the oviducts and then digest them with the peptidase, and then collect the eggs with a mouth pipette and wash them several times till you get all the junk that's on them and they're nice and clean. And then this is what it looks like. There's our two pronuclei. This is our holding needle, and then this is our injection needle over here. And so then you put the needle into the pronucleus and inject a few nanoliters in there, which swells it about twice its size if you get the right amount into the pronucleus. And then the next, excuse me, the next stage is to transfer the fertilized eggs back into the oviduct. So this is your ovary back here. You can't really see it, but this, the oviduct is just a really tightly wound up tube. And so you take a needle and make a little slit in the tube, and then put your transfer pipette into the oviduct, and then you just give it a little blow, and your eggs go into the oviduct. Okay, so I want to talk about mice that I worked with a long time ago. But it's such one of the best stories there is about genetics and how we're able to decipher a biochemical pathway with just transgenes and knockouts. And so agouti is a coat color gene in mice, and you can have the mouse on the left has a complete loss of function in the agouti gene, so it's jet black. And then you can have the one facing away from us is a wild-type mouse, and so it's part brown and part yellow hair, and that's where you get your camouflage from. And then, of course, you can have an overexpressor or a mutation in the melanocortin receptor to have a yellow mouse. And then these mice have been bred for centuries and stuff, and you have all different kinds of combinations that the mouse fanciers have. So there's the real mouse, the real agouti rodent. It's a South American rodent, and it has the typical wild type color. And where this happens is that during the hair growth cycle, okay? Normally you start off, you have black pigment, or actually start off here, you have black pigment being produced in the melanocytes, right? And then about day 4 of the hair growth cycle, you have this burst of agouti expression, okay? And then after day 5, agouti not expressed anymore, okay? But what we were interested in was what's been called the yellow obese syndrome. So you have some mice that have obesity, they're hyperglycemic, hyperinsulinemic, they have increased incidence of tumors, and they're also bigger. And so we wanted to try to know what was causing obesity in these mice. And when we did a northern blot, we find agouti not expressed anywhere in the wild type mice, except this little bit that's smaller, and it's only in the day 4 skin, okay? But in our AY mice, we see it expressed at really high levels in all tissues. So our question was, once we cloned the mutation and discovered what was going on, what's happened is there's a gene called Rally, okay, that's upstream of agouti, and then you have a lot of different 5' splice variants for agouti, and then the 1, 2, 3, and 4 is the open reading frame, so that's where your protein comes from. So what has happened in this lethal yellow mutation is that all of Rally has been deleted, and all of these 5' non-coding exons are deleted, and the 5' non-coding exon of Rally splicing into the open reading frame of agouti. So that's why you have this. Normally, Rally is expressed at high levels in all tissues. So you have Rally's promoter driving agouti expression in all tissues. So, but the question was, what's causing the lethality and what's causing the effect? Because we know we've disrupted two genes, the Rally gene and the agouti gene, right? So what we decided to do was make a transgenic animal, and we used the beta-actin promoter, which is expressed ubiquitously, to drive only agouti expression. So now your Rally gene is going to be fine, and agouti is going to be expressed at high levels in all tissues. And what you see is the transgenic males and the transgenic females in red are fat. And they're also yellow. So that tells us that agouti alone can cause the obesity and the changes in hair coat color. And we also went in and showed that it was really Rally that was causing the lethality too. So, but at the same time, we cloned agouti in humans and found out that it's expressed at highest levels in humans in the adipose tissue. So we made a tissue-specific overexpressor of agouti by using the AP2 promoter, which at that time we thought it was only in the adipocytes, but now we know it's in the adipocytes, macrophages, and a few other cells. But that was state of the art back then. And so what we decided to do was humanize the mouse. So we took the human cDNA and overexpressed it with the tissue with a fat-specific promoter to see if that was contributing to the obesity. And as you can see, that the transgenic guys, when we overexpress the agouti in adipose tissue, are fatter than the wild-type mice, with no differences in fat-free mass. And so now if we look at our three mouse models together, we have the non-transgenic, and then our AP2s are specific for adipose tissue, and then we have the beta-actin agoutis, which is expressed in all tissues. So the expression in adipose tissue is causing some of the obesity, but not nearly as much as the obesity that's coming from another place. Okay, so about that time, some people at Amgen cloned the agouti-related protein. Okay, like agouti, it's expressed in the hypothalamus, which of course all of you know is important in body weight and energy expenditure. It's also expressed in the adrenals at quite high levels too. So what they did, they made two transgenics. One that overexpressed AGRP, so they got a black fat mouse. And then like we had done, they overexpressed agouti and they had a yellow fat mouse. And so I had to take kind of some of the next slides out, because of the newer technology that I'll present at the end. You can't go through the whole story. So it turns out that HERP binds to melanocortin receptor 4 in hypothalamus, and that regulates food intake. And the agouti binds also to that receptor. It binds to all the melanocortin receptors. So not only is it binding to MC4 in hypothalamus causing obesity, but it binds to MC1 in the skin, causes co-called phenotype. Any questions on that part? So it's just a quick example of overexpression and tissue-specific overexpression. Wow, it was 1997. That was a long time ago. So, also, Mitch Lazar's group cloned this gene called resistin that's produced by adipose tissue, and they were able to show that it was associated with insulin resistance, and I think in some cell lines, the added cell lines that caused insulin resistance. So we didn't want to make a mouse that had insulin resistance from birth. So we wanted to use a inducible tissue-specific system to wait till the mice were adults and then induce insulin resistance in them. And so this requires a 2 transgenic system. So we have, again, our AP2 promoter, okay? And it's driving the expression of a protein called RTTA, okay? It's a mutated tetracycline binding protein, okay? And then this other mouse we made has a tetracycline inducible element, and normally resistance there, but it's not expressed. But when you cross these 2 lines together and you have both transgenes, they still don't produce resistin, but if you add doxycycline to their drinking water or their food, then doxycycline binds to the RTTA, and then it binds to the tetracycline response element, and then you have resistant expression as long as you have doxycycline in the water. Okay, so, and what we were able to show is that if you have both transgenes, right, and doxycycline, then we could take their blood glucose from 300 to 500. And, but if we either had one transgene or the other and added tetracycline, we didn't have any significant increases in blood glucose. So that was an example of an inducible tissue-specific promoter. So that's about it for the overexpression part. So any questions or anything? Okay, so now we get to the more sophisticated stuff. This is the gene knockout approach. So again, you can do a knockout and a whole body knockout, and a lot of times that is an embryonic lethal. So you want to be able to do a gene-specific knockout, or again, an inducible gene-specific knockout. And to do this gene-specific knockout, you want to, most people use the Krelok system, which I'll get into a little bit. All right, so somebody says, Randy, I want to make a mouse, and how long is it going to take, and how much is it going to cost? And I said, well, if we're lucky and everything goes as planned, you're probably looking at least, a year before you even have any mice to breed. And then you've got all these different cycles of breeding, which is going to take you another year or so to do that. So a year and a half, 2 years is a good timeline for doing that. So, decide to make a mouse and write a grant on it, and so I can't get my grant because I don't have the mouse. That's just not the way it works. So, but I'll go through each of these steps that are necessary. So, the first thing is you have to have the DNA to start off with. And so, if you say, okay, I want to knock out this gene, we'll go to a bacterial artificial chromosome library that has, you know, just the entire mouse genome in it, or whatever species that you want. And then we want, of course, just a small section of that entire section of the chromosome. So we'll retrieve, I won't go into all the details and stuff, but anyway, we will retrieve that piece out of the back. And then we will insert the LOXP sites where we want them in the genome. And then linearize the DNA, electroporate it into the embryonic stem cells, and then screen the ES cells. And what I've said in just 30 seconds takes Jin Ying probably 6 months to do all that. So it's if everything goes great, it's 6 months. So the way we make these constructs is, this is not to scale, but after we have our vector, right? On each end of the vector, we will have about 5 kilobases of homologous DNA. So it matches exactly with the gene you're trying to knock out, right? So when you do that, then you have homologous recombination between the two similar ends of DNA, and you have 2 selectable markers. One's in your engineered construct, so that's like a puromycin or something like that. So you can only select cells that have your insert. But you can also get random insertions where it's not homologous recombination. In the case of that, you have a negative selection marker, usually it's thymine kinase. Usually it's thymine kinase. And so after you treat with puromycin and you treat with both of them at the same time, then you'll kill off all the mice or rats or eggs that have the transgene inserted or the non-homologous recombination. So this is more or less just what I went through, just in a little more detail, showing the details of a gene and how we have our homologous ends and then our selectable marker, and then you end up with your 2 LOXP sites inserted wherever you want. And then how LOXP works is once you have your 2 LOXPs inserted into a piece of DNA, you cross them with a mice that overexpresses Cree recombinase. And so what the Cree recombinase will do, it'll recognize this LOXP site and this LOXP site, and then it'll clip everything between them and take half of this LOXP and half of this LOXP. Then you have only 1 LOXP left, and everything that's between them is gone. Again, you could have a cell-specific promoter, driving your Cree, and then you have your LOXP, your target gene, and then you only delete your target gene in a specific cell type. So what happens once? That's a little too detailed one. So once again, we go back to our chart of embryogenesis, and now what we have is we have our blastocysts, right? And then you have your blastocele cavity, And these are all embryonic stem cells, okay? And what we want to do, so these are our targeted embryonic stem cells, and you can see them in our injection pipette, right? And then we will take those and inject them into the extracellular mass that contains the endogenous ES cells. So you have a mixture of your targeted ES cells and a mixture and your endogenous ES cells. But what I didn't say is that our targeted ES cells come from an albino mouse, and the blastocyst is from a black mouse. So when you have your pups born, first we take again so this time, since the eggs are further developed, you know, when we had our pronuclear eggs, we implanted them into the oviduct. But now that we have a blastocyst, we'll make a small cut in the uterus and then implant the eggs into the uterus. And this is a lot easier. So now, okay, this is the black. So we had our ES cells were from black males, and our albino blastocysts. And so then this is what you would want to get, but it's not quite that pretty. What we get is this. So you just get this, you know, completely dark mouse to, you know, one that just has black whiskers, and it's a totally random process. And so usually we want to take your black, the ones that have the most black coat color, and breed those to another group of albino females. And if you get black pups born, right, then you have germline transmission. And the reason we do this is that it's not necessarily any correlation, but we think if you have a higher coat color from chimeric cells, then you have a chance of having a sperm that came from a black 6, from a targeted cell. So unless that particular sperm that fertilizes the egg came from an ES cell that you injected, then you won't get germline transmission. And that is the biggest problem for this. This is because there's only a few mouse cell lines that work this well to go germline transmission. There might be one rat ES cell line that works this way, maybe a few pigs and a few cows, but you know, it's just, you can't do these genetic manipulations in other species simply because you don't have ES cells. And if you're going to do a cow, then you've got 10 years before you can, before they're mature and breeding and germline transmission and stuff. So we'll talk about the CRISPR a little bit later. So another thing that we have to do is when we make this construct, right? So here's this would be like we have a mouse, and we're genotyping it, right? And so here's exon 4 is what we really want to get rid of, right? Ultimately. But when we make the construct, we have to put in your beta-actin selectable neo markers, and then this one particularly has some other beta-gal markers, so you can see what cell type your gene is expressed in. But the thing is, this is a huge segment of DNA that's stuck right in the middle of the gene. More than likely, it's going to cause some type of aberrant expression of that gene. So you need to get rid of it. All this big chunk, so this is another recombinase called FLIP, FLP, and it recognizes these FRT sites. And so the first thing we do is breed it to a beta-actin FLIP mouse, and we remove this. And so we went to like 5 kilobases down to 18 base pairs, which still has a probability of screwing something up, but not nearly as much as that big piece. And then you cross with your Cree, right? And then you take out exon 4. And what almost always happens by design, it's uncanny, that usually when you remove an exon 3 will splice into exon 5, right? And then you can have the rest of your gene transcribed. But almost always it creates a stop site, a mutation very close to where the splice site begins, and so that's rapidly degraded and never makes protein. So that makes our life easier, but it can happen. And then this is just the breeding strategy, going through ultimately when you start off with your 2 mice, you go through all these different lines, and finally you get down here to 1 out of 14 after 3 generations has the right genotype that you want. So then you have to start breeding those 2 guys together and to generate your extreme ones. You know, and each round of that is like 8 weeks. So another thing, you've probably heard of knock-ins, and for a knock-in, you kind of have the same approach, except you have your exons, right? And then you have a splice acceptor, and then a reporter gene, okay? And this reporter gene can either be like beta-gal, or you can stain tissues, you can overexpress fluorescent proteins, you can overexpress toxins that will kill any cell where it's overexpressed. And then again, you can have your Crees and your FRTs. This is the usual strategy for making all of those lines. And this is just one line. This one overexpresses green fluorescent protein. And one of the promoters is the one on the left is a pancreas-specific promoter. So you can see that it really lights up the pancreas really nicely. And then this one over here is kind of a neuronal promoter. I don't know if you've heard of the DREADs, but these are designer receptors exclusively activated by designer drugs. So these are mutant receptors. They're the mutated muscarinic receptors, and they have these coupled to, you know, MD3s. It induces neuronal firing, okay? The MD4 silences neuron signaling, and MD5, and then this one you can get it to increase cyclic AMP if that's what you want to do. So you have this clonazepine inoxidized, CNO, right? And you have your transgenic mouse that has these whichever one of these designer receptors in it, right? And then stereotactically, you can inject your CNO into a specific area of the brain and either activate the neurons or silence them. And so without going into a lot of it, so here you have your synapsin promoter driving your MD4, which is an inhibitor of signaling, And you also have the green fluorescent protein in the construct as well. And so what this allows you to do is you can find the green fluorescent protein and then put your recording needle on that specific neuron and do direct one-cell recordings from a specific neuron. And as you can see here, these neurons are firing at about 40 times a second or so. And then they add the CNO, right? And you just completely block firing of that particular set of neurons. And so you can record from it if you have it in a slice, but also you can look at feeding behavior or activity. And so everybody thought, oh, this is great. You know, it gives us a really specific tool that's going to really help us define these particular sets of neurons. Right? So about 3 weeks ago, a science paper came out where the body takes the clonazepam in oxide and cleaves it to clonazepam. So clonazepam is an antipsychotic drug that works on a lot. It's really dirty and it works on a lot of receptors and stuff. So all this, it's down the drain right now. So I don't know how people are going to interpret their observations when now that they know that you're actually putting clonazepam on the neurons. So the other is optogenetics, okay? So I'm trying to get some of the newer stuff in at the expense of showing you more models and stuff that we make. But with the optogenetics, what you have is, again, you have your promoter driving expression in the specific neuron, or it could be in all neurons. It doesn't really matter for this case. It could be in the brain, right? And then you have your gene encoding opsin, which is a light-sensitive ion channel, okay? So this way, in this particular mouse, they injected a virus, let's say, into the third ventricle, and so now you have this opsin expressed in that area of the brain, okay? And then you implant your optic fiber into the specific area that you want to activate, and then turn on the light, the laser, and it activates this opsin channel, and calcium comes in and the neuron turns on. And then once again, you can look at whatever behavior you want to study. So that one's pretty cool, and a lot of people are using that as well. The other one that I'm not a big virus person. I like to make the mouse, and you can use it over and over again and stuff. So one thing you can do is you have this Rosa 26 locus. You can knock it out, and there's no phenotype that anyone's discovered yet. And so you can have your opsin DNA again, but what you have is you have a transcriptional blocker, okay? So as long as this transcriptional blocker is between the opsin and the promoter, then the gene is silenced, okay? So, but once you remove the transcriptional blocker by crossing it with a crene line, which would be a neuronal specific or whatever tissue you want, then you have opsin cDNA produced, and then this is just a genetic model rather than virus. And then again, you can expose that specific neurons to light with the laser and get the same activation. So as I said earlier, we're talking about, you know, it's going to take us 12 months, and then it's going to take you guys, you know, another 12 months to a year to get your germline transmission and experimental animals come. And it's going to cost about that $10,000 to $15,000 if we do it. So now, this CRISPR comes along that about 2 years ago that, you know, it's going to really speed the world up. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Okay? And I really don't know what that is, but the Cas9 is a endonuclease that makes double-stranded cuts in DNA. Okay? So here's the schematic of it. So you have your guide RNA, okay? And this guide RNA matches your gene of interest, where you want to cut that gene, all right? And then you have your Cas9 protein, okay? And so this loop in your guide RNA interacts with your Cas9 protein, and then it finds your target sequence in the genome, and then makes a double-stranded cut right at the end of your targeting sequence. You know, right here, it makes a double-stranded cut. And so your genome does not like to have double-stranded cuts in it. And it tries to repair it just as quickly as possible. And so one way, so here you have your Cas9, and you make your double-stranded cut, right? So the quickest way for it to do this is the way it normally repairs double-stranded cuts is non-homologous end joining. So it really grabs any nucleotides that are available and repairs that gap junction, that two-stranded cut, okay? And it's not very precise. So what it usually ends up doing is making a point mutation, just like we did when we did our splicing out our exons, a point mutation that causes the gene to be silenced. Okay, so this is just the brute force, knock out a gene in every tissue, very fast. The other way is to do homologous directed recombination. This is where it's pretty powerful, because you can recognize that there's a gene with a mutation in it, right? And that gene causes blindness, other disorders, and you can repair. So what you do, just like we did for homologous recombination in our ES cells, we have our mutated site right here in the genome, and then we make this vector that's got the mutated site repaired, and then you have the homologous domains on each side of it, and so you can insert this repaired piece of DNA into your target gene. And it works pretty well as long as it's a short piece. We've tried putting longer pieces, doing log speeds, the whole thing, and it's just not efficient. So we've went back to using our embryonic stem cells for the large fragments. Once again, what are the advantages? Of course, it bypasses this entire process, everything from here up. You're making your vector, you're targeting your ES cells, the injections. So what you can do is you can order this target DNA for about $50 and get your Cas9 protein, inject both of those into the pronucleus, and you're done. And you get your gene knockout. So that takes about 2 months versus 12 months. So it's really time efficient and cheap. So again, site-specific germline integration, it takes 2 to 3 months. And the other beauty is that it doesn't have to be a mouse cell. They can be rats or monkeys. So people have been CRISPR monkeys, cats. Sangley makes CRISPR cats and crickets. Apparently they're good for some respiratory model. But it opens up the possibilities. So we decided to use CRISPR to knock out uncoupling protein 1 in rats. So uncoupling protein is part of brown fat and, that allows uncoupling of oxidative phosphorylation. So you just create a lot of energy and waste energy. And so we know with mice that they, if you knock out UCP1, you put them in the cold, they'll lose about 4 degrees Celsius per hour. So it's pretty rapid, and you have to remove them. They can't take it in acute cold exposure. You can't cold adapt them if you just lower the temperature gradually, like 2 degrees every day, you get them down to 4 degrees. But they can't take an acute cold exposure, and they have such a small surface area, and so we wanted to make the rats. They're 10 times larger than mice, and have a more similar physiology to humans than mice do. So we thought it would be a better model. So once again, we made our UCP1 guide RNAs and Cas9, did our injections. And we wanted to target exon 2. So that's where our guide RNA was. And then after we had our rats born, we started to genotype them and look at them. And this is really what we expected to see. Maybe like a 5 base pair deletion in exon 5, that right there, specific exon 2, right where we made the cut. But you see the second rat, it had so many mutations that we couldn't even genotype it. So it was just, it had cut everywhere. And then our third rat, it had part of exon 2, part of intron 2, and so it was just kind of, it made the cuts there, and they all produced a knockout, so for our purposes it was fine, but as you can see, it's not quite as accurate as we thought it would be, or as it was billed to be. But what happens when we put the rats in the cold? And this is acute cold exposure. Both the knockouts and the wild types lose a little bit of body temperature, but they're clearly able to maintain body temperature above 36 degrees, and they can do it up for 2 weeks if you leave them in here. So they're perfectly fine at cold adapting. But they do things like we do when we put them in the cold. The knockouts are in the black bars. By 6 hours, there's just one of these guys that started to shiver, right? And then by 8, 10, and 12 hours, you get close to 80, 90% of them shivering. And they really do. I mean, they're just that. It's just like you when you get a cold chill. They're shaking. And it's quite clear. It's not just a little subtle thing. And they continue to do that for about 8 or 9 days. And then finally they get the induction of whatever is induced. It's not UCP1, but they get some other type of thermogenesis upregulated. And so I think that's why it's a good model, because we could uncover some more thermogenic pathways that we have, that since we don't have a lot of UCP1. But they also eat more too. So they increase their food intake, which is the thermic effect food in the shade. So they're staying warm that way, rather than shivering. Rather than UCP1. And then the last, I could have kept some stuff in, but here's the bad part about CRISPR, which is really scary. So there's this RAD1 mouse, it's blind, right? So they knew the point mutation. They replaced this mutation in the PDA6B gene with the phosphodiesterase. And they cured blindness in the rat, in the mice. It worked, and you know, you can do that, that's good, right? Well, the bad thing is that these guys decided that they would sequence the entire genome of 2 lines of these mice that they cured blindness in, right? And what they found was that, and these are all different methods of analyzing the data, they found that in line 1 they had 1,700 single nucleotide variants. Okay, so in that mouse that had over 1,700 mutations created by the Cas9, right? And in the second line, it had like almost 1,700 again, right? And when they analyzed both of them together, 1,300 of them were the same mutation. So this is, you know, it's just not randomness. This guide RNA is finding different places in the genome to edit. Yeah.
Are these mostly insertions and deletions, or are they?
These are single nucleotides. These are the indels here. In the bottom, indels are the insertion deletions. And so in those mice, you know, they had 164 in one, 128 in the other one, and 170 in those were in common. So, you know, I don't think that's a good. It may just be because with this particular guide RNA and the point mutation where it was, they're always ranked in terms of specificity and stuff, but if that's the only choice you have to use that one. And if it's very non-specific, you're going to get a lot of off-target hits. So, so once again, there's another thing we think is going to be great. Change everything with the dread receptors and stuff that may not pan out to be quite so good. But I will say this. You know, I get an email every day for a new improved CRISPR, new this, new that. So they're trying to sell you stuff anyway, whether it actually works. We have Gene does the gene targeting, Sangley does the injections, and Trelly does the cell work. And the thing, like I said, it's a turnkey service, so you just come to us and say, we want to knock this gene out or do this, and we will do it. We're getting close to 70 now, and so far we've got all of them to work, some of them not as quickly as others, but it's working. That's just what I do for fun. So questions?
Thank you for that.
Yeah.
So when you look into the future,
Right.
what do you see in terms of being able to apply some of this to human?
They said they've already started some, what is, I'm drawing a blank, but neuromuscular disease.
ALS?
No.
Multiple sclerosis?
Yeah, they've already, are doing some trials with multiple sclerosis in humans. And it'll happen, you know, somewhere else before it happens in the US, obviously. But, yeah, it's kind of, you know, if you take cells and you correct them and then properly make sure there's not a lot of other mutations and then replace the population with those, then that's probably a lot safer. And, you know, there's I can't think of this gene either right now, but if you have this gene, you will not get HIV. Period. You're completely resistant to it, so.
It's pretty interesting.
Yeah, and, so if you do a routine mutation of that gene as a vaccine or something, in embryos or whatever.
So can you would you take a guess how far down the road in the next decade?
I don't know,
Would you-
Before 5 months ago, I said, oh, it's going to be really quick, but then all these off-target mutations and stuff, I don't think I wouldn't want to use it unless I was on my way out,
I don't know whether you want to answer that, but what's your take on genetic manipulation, embryonic development in humans?
I don't know. I would try not to think about it. I don't think in most cases, it's probably not ethical. But I think if you, yeah, I know, if it's a specific disease and you can treat it and be effective, I would see why it shouldn't be done. But to, mutate MILD or something, have double muscle people or something. I don't think that would be ethical.
How do you think that those techniques are going to improve the production of animal for food? I mean, it's going to be better?
I think you can. I think, the drawback's been not having the ES cells. But I'm sure you could, the technology's there if you just know which genes to target. We're all GMOs.
Yes.
And everything we eat is, so.
Are there any standard methods that are used to verify whether or not a knockout or knock-in was successful, or do you just kind of?
No, we will, we do as much as we can to make sure there's not off-target effects with the screening process. And then once we have the mouse, then we will go in and sequence that entire fragment of DNA and what's on each side of it and everything to make sure everything's exactly like it should be. And so used to we'd do southern blots, and you just look at patterns and things. But this way we're sequencing it, so you know it's there.
Let's say that CRISPR did work, you didn't get all the mutations. What would be the next step in pressing this, moving it forward?
It's so fast and easy. Research-wise, it would speed up the number of genes that you can mutate easily. And then, once again, replacing point mutations in certain animals and other species. I think the other species is probably as important as anything, because mice aren't just, my boss at Oak Ridge used to say they were just very little people, but they're not. They're a lot different than us. But I think, the particularly primates and pigs.
So is it, this question's kind of out here, I guess, but is there anything in place for humans if someone were, for instance, pregnant and knew that there was some disease?
I don't know.
And was open to some kind
Yeah,
of
like I said,
manipulation?
it'd have to be extremely experimental with a life-threatening condition or something like
Right,
that.
yeah, but there are those.
Yeah, they are, yeah, without a doubt. And so, I wouldn't be surprised if there's places outside that aren't as tightly regulated as the
Right,
FDA,
okay.
and probably doing stuff like that.
That's pretty amazing.
That really is.
That's pretty amazing.
I mean, how far it's advanced in 2 years, you know. There's lots of Californian projects
So
to
in what are
do.
you working right now? In what project? Are you
So-
working right now?
On,
Are you working on a specific project right now?
oh yeah, I mean, we've got like, I have my own that I'm doing. And then there are, I guess, about 10 or 11 different ones in the queue that we're doing. So, yeah, it's a continual, process, so we have to keep our production going and charge people and defer part of the cost.
So what's the potential for using the technique for increasing food? Or the kind
Yeah,
of...
I mean, it's the same way, yeah, I mean, I think you can, I mean, people have been modifying plants for better food production and disease resistance and certain, I saw that one pesticide resistant corn and soybean that they have that they're using. And the pesticide was drifting off and killing everything else. But
And what about animals, more
you see quite a bit of stuff
Production.
with pigs.
with pigs?
Yeah,
Okay.
yeah. Yeah, the transgenic pig, they've been around for quite a while. And sheep as well.
There's cows too.
Cows, yeah, yeah. It just takes a lot longer. But the technology's there.
What are we usually shooting for as far as the guide RNA limit?
I use just 19 base pairs.
Only 19 base pairs?
Yeah.
Okay, well, that's why there are so many off target.
Yeah, I guess, but if you take, if you assume, which is it's not entirely random, that you have a 1 in 4 chance, 1 in 4 base pairs, right? So that's 4 to the 19th power, which is a pretty big number. if it's all just random.
Yeah,
Yeah,
the
right.
primers, DNA primers are about that length, and they're not too,
Yeah, so,
they don't create too many off target. So-
right, exactly, so either it's 2 or 4, but either way, it's a pretty big number.
Any questions, comments?
But it's not random, that's the problem.
Yeah.
You'll have something that's common with a lot of others, areas in the genome.
Well, thank you. Very
Sure, I
interesting.
enjoyed it.
Very interesting.